One of the most important aspects of scientific research, and understanding the world through a scientific perspective, is the use of models. Models are simplified systems that represent or mimic real-world systems. And by using models, we can make controlled changes or reconfigurations to alter the system. And by studying the outcomes of those changes, we learn more about the system itself. And of course, as we learn more about the system itself, we can refine our models and make more predictions and more sophisticated tests. For example, some of my personal graduate research involves mice that have been selectively bred to exhibit behaviors that model the symptoms of a particular psychological disorder. By testing drugs with these mice, we can find potential candidate therapies for humans with that psychological disorder. Mind you, a lot more testing is involved downstream from me to ensure drug safety and efficacy and all that stuff. But experimental drug testing has to start somewhere, and the animal models are how we do it. Anyways, the news story that I bring you today involves an unprecedented computer model of a cell. Now, this might not sound too crazy, but let's get into some of the details, and you'll soon see why this is so impressive and potentially groundbreaking. First, the University of Illinois scientists wanted to get a real-life organism to base their model on. They used some of the simplest known cells, belonging to a parasitic bacteria known as mycoplasma. Earlier research with the mycoplasma at the J. Craig Ventnor Institute has trimmed down the microbe's genome, cutting out unnecessary genes and leaving behind only the bare minimum, a skeleton crew of some 500 genes that could keep the most basic cellular processes running. For comparison, E. coli cells use around 5,000 genes, and any given human cell uses around 20,000 genes. Now, with this much more manageable 500 genes to work with, the scientists then began programming their model. In addition to these 500 genes, they also included almost 150 metabolites, more than 450 proteins, and thousands of reaction schematics and biochemical pathways. With all of this data loaded up into the Lattice Microbe software operating on the NVIDIA Tensor Core GPUs, the researchers created a cell model with approximately 2 billion particles and initiated their three-dimensional cell model program for a 20-minute simulation. I should take this moment to point out that they defined each simulated particle as an atom, and each particle is actually representing a molecule that's composed of atoms. So it's more like 2 billion molecules, not 2 billion atoms. It has to work this way because the amount of computations involved can quickly become completely unmanageable. It probably took days for their computers to process this 20-minute simulation with 2 billion molecules. If they actually wanted to simulate 2 billion molecules worth of atoms for 20 minutes, and keep in mind, if they were just simulating a bucket of water with 2 billion molecules, that would be 6 billion atoms. Now imagine doing that for a cell with all sorts of different molecules of different shapes and sizes, including some that have dozens if not hundreds of individual atoms within them. If you were to try and simulate that for 20 minutes, the computers would be processing data for decades. Anyway, what they saw in their simulation was amazing. For one, the cell survived. This computer model, at a fundamental level, worked. The simulated cell did not collapse, it didn't die, and this suggests that the basic mechanisms of life are being sufficiently represented in this model. Furthermore, novel emergent behaviors, stuff that wasn't explicitly programmed in, began to appear. The simulated cell underwent complex metabolic processes like changing the rate of consumptions of amino acids and nucleotides to optimally manage them despite a limited supply. They also saw the emergence of enzyme complexes, or at least parts of them, and the cell actually grew larger, appearing to ready itself for replication at a rate that's really pretty similar to your average single-cell replication rate. The authors note many similarities between their simulated cell and real-world cells, including average metabolite concentrations over time, the surface area doubling time, the mRNA half-lives, and the energy that was spent on transporting stuff across the membrane, among many other things. But the model wasn't perfect, and the authors noted several discrepancies between their model and real-world cells. But at the end of the day, it's an incredible simulation, an incredible advancement. Now that we have this working model, we can start using it to test stuff and learn more about the nitty-gritty inner details of the cell and life in general. We can also add stuff to it, making the cell more complex and more lifelike. And with more powerful computers, we can simulate the introduction of drugs, or poisons, or hormones, or anything else, and use this model as a basis for clean, rapid computational experimentation. Read through the paper, and you'll see that a lot of this stuff is really remarkably technical, both in terms of the computer science that's way beyond me, and in terms of the biology, a lot of which is also way beyond me. But it's definitely one of those ultra-valuable developments that will likely be used as the basis for groundbreaking discoveries in the near future.